Hello, I'm Markita Landry. I'm an assistant professor in the Department of Chemical and Biomolecular Engineering at UC Berkeley, and also a 2016 Beckman Investigator. Today I'm going to talk about some of the developments that my lab has been working on over the past 4 years, centering on the use of nanotechnology to engineer solutions to life's big problems. So my lab largely focuses on understanding the structure and function properties of nanomaterials for various purposes. Nanomaterials are nanoscale counterparts to their bulk material forms and can often take on unique properties when confined to the nanoscale, such as unique optical properties or unique mechanical properties. So specifically, we exploit some of the unique optical properties of semiconducting single-wall carbon nanotubes to develop optical probes that we can insert into the brain to image brain chemistry. Now, brain neurochemistry is an important feature, as I'll talk briefly about today, of understanding the brain in both health and disease. And these probes can be inserted into the brain extracellular space to understand how neurons communicate chemically with each other as they shuttle neurotransmitters back and forth. Another interesting feature or property that nanomaterials can take on at the nanoscale is exceptional tensile strength and pretty extreme aspect ratios. Now, these properties lend nanomaterials well for delivery applications, where it becomes important to both surface functionalize and controllably graft biomolecular cargo for delivery into living systems. Now, the systems that we focus on predominantly are systems of agricultural relevance, specifically the delivery of DNA, RNA, and protein into plants. So towards that latter aspect of the lab that I talked about, the delivery of DNA, RNA, and proteins to plants is fundamental for the ability to genetically engineer or genetically manipulate or edit plant systems. And the reasons that we would want to do this are manyfold. So first of all, in agriculture, plants can be conferred with desirable traits through small-scale genetic manipulations that allow them to be more robust to a changing global climate or to pest or pathogen infestation. In the pharmaceuticals industry, plants are often the source of precursors or of natural product-based drugs. And so being able to manipulate biosynthetic pathways that allows upregulation of these naturally occurring small molecule therapeutics is advantageous to the pharmaceuticals industry. And lastly, in the bioenergy sector, being able to genetically manipulate plants can make certain crops more efficient and allows us a genetic handle with which to improve biofuel production from certain crops. As I mentioned before, all of these endeavors involve the delivery of DNA, RNA, or proteins from outside the plant cell to inside the plant cell. And this is a challenge that's been taken on by two group members, Ener and Zhang. Now, current methods for delivery of biomolecules to plants, and I'll focus specifically on DNA as kind of the genetic workhorse for a lot of these endeavors, are twofold. So first is agrobacterium-mediated delivery. Now, with agro-mediated delivery, we're taking advantage of a plant pathogen interaction and manipulating this bacterium or agrobacterium at the bench to contain DNA that contains our gene of interest before that agrobacterium can then infect plant tissue, thereby transferring our gene of interest into the plant cell and incorporating that gene into the plant host genome. Now, agro-mediated delivery has advantages in being extraordinarily efficient. However, because this is a biotic method of gene transfer, there are species limitations, whereby it might be relatively straightforward to use agro-mediated delivery for genetic transformation of certain easily manipulated crops or model plant systems, but can be more difficult for their wild relatives or non-model counterparts. Additionally, agro-mediated delivery is only limited to delivery of DNA. So if we want to deliver, for example, small interfering RNA or proteins, the preferred mode of doing so is through biolistic particle delivery, which is cargo agnostic. Here, what we can do is dehydrate the cargo onto the surface of gold particles, and using a high pressure gene gun, quite literally shoot these cargoes into plant cells. The plant cell will then incorporate some of this DNA, RNA, or protein, and in the case of DNA, there's often integration into the plant host genome as well through endogenous repair mechanisms of the cell. Now, while this methodology is more cargo agnostic, it also is accompanied by fairly significant cell damage and low transformation efficiency. And in both cases, we have a lack of control over the frequency and location of transgene integration. What this means is that, at least within the United States, the classical definition of a genetically modified organism, or a GMO, is going to be the integration of foreign DNA into a plant genome that is then intended for human consumption, or the use of a pathogen such as agrobacterium in the production of that genetically modified transformant. Now, despite the drawbacks of the technologies that we currently have, we have had many successes in the development of genetically modified crops. Here I'm showing the USDA approved genetically modified crops, some of which are intended for human consumption, some of which are not. But as you can tell, the United States produces actually quite a large number of genetically modified crops that are currently approved and field produced. One of my favorite international examples is the generation of a varietal of rice called golden rice. Golden rice is a type of rice that has had the biosynthetic pathway for the generation of vitamin A incorporated into it. And as you can see, the varietal of rice known as golden rice therefore kind of glows yellow with this, the expression of this vitamin. And the concept here being that rice being a staple food in many countries where the population is largely vitamin A deficient can help by allowing this, this vitamin to be abundant through the food supply through a genetically modified crop. Now, again, despite many of these advances, the production of a genetically modified crop today comes at a significant cost, specifically on average 13 years of research and development and a cost of $136 million on average. And about 40 of these $136 million are regulatory spending. And so in my lab, we sought to develop a technology that would address the low efficiency of certain existing tools and the species dependence of biotic tools in developing a technology that is efficient, but also one that could potentially be non-pathogenic. So an abiotic tool that would also allow better control over avoiding transgene integration of the delivered DNA cargo. Now, to do so, we had to think a little bit about what it might take to accomplish these goals, specifically a force-independent and pathogen-independent delivery mechanism for DNA into plant cells. If we look at the membranes that are limiting the delivery of molecules from outside to inside the cell, we'll see or we'll note that the plant cell, as with all cells, has a lipid bilayer. Now, the size exclusion limit of the lipid bilayer, or the size of something, the maximal size of something can be before it can no longer traverse this membrane, is about 500 nanometers. If we look at the size exclusion limit now of the plant cell wall, we see that that size exclusion limit drops down to about 5 to 20 nanometers. What this means is that the dominant barrier for delivery in plant cells uniquely is going to be this very small plant cell wall. And so we thought a little bit about the form factors of what we might want to use as abiotic carriers of DNA for delivery into plant cells. And if we think a little about the form factors of, for example, carbon-based nanomaterials, we'll see that zero-dimensional nanomaterials are small enough, but the surface area available for biomolecule grafting is quite low. Conversely, two-dimensionally extruded graphene, atomically thin, however, now with no dimension that's below the size exclusion limit, provides surface area, but does not stay below the size exclusion limit. And so we're predominantly focusing on high aspect ratio nanomaterials, ones that are small enough in their smallest dimension, but also have an extruded first dimension that provides surface area for biomolecular grafting for doing chemistry. So the concept here being that we could attach cargo, such as DNA, that this DNA would be delivered through the plant cell wall and the cell membrane into the nucleus, where the DNA could be transcribed and translated into a reporter protein, such as green fluorescent protein, allowing us to quantify the efficiency of our platform. And so the tests that we've done so far are ones that allow us to graft biomolecular cargo onto these nanomaterials and introduce them into mature plant tissues to see if we observe transgene expression. We can do so by infiltrating the underside of leaves and allowing a few days for transcription and translation to occur, and then taking a confocal micrograph to assess at first pass whether green fluorescent protein is being expressed. And so as you can see here, for various plant species, we observe green fluorescent protein expression in tobacco, arugula, wheat, and cotton. And the expression profile that we see is largely through the entire leaf thickness. Now, an interesting feature of this platform is that while we observe maximal expression at day 3, by day 10, both with confocal microscopy and also as measured with qPCR, we see that the expression has largely subsided, which we've now validated is due to the transient expression nature of the delivery platform, which means that there is no integration of foreign DNA into the host genome. Now, this is a platform that I think would be most exciting to use for a different or in combination with a different technology, specifically CRISPR-Cas nucleases. The concept here being that we could deliver a plasmid that codes for the CRISPR protein along with a guide RNA, that this system would be expressed once delivered into a plant, and that this CRISPR complex, based on the guide RNA sequence, would edit a site-specific location on the plant host genome. And if this edit is a knockout, we would get the plant genome modified plant tissue in the absence of integration of the transgene. And this is especially important for crops that are known as vegetatively propagated, in which crossing out transgenes such as CRISPR would not be possible. So our lab is excited to work in different plant tissues as well, so that we can regenerate whole plants that would have these edits incorporated into them, having advantages both on the regulatory side and also on the precision of where we're making these edits. Now, shifting gears briefly so I can talk about some of the work that we've been doing in developing probes, not for the delivery applications that I just spoke about, but for imaging applications. Now, the applications that we're most interested in here are in the neuroscience space. And the reason that we're interested in developing probes for brain chemistry is that the brain is composed of a very multifaceted set of different properties. First of all, there's structure and connectivity, so basically the anatomy of neurons and where they are in the brain. The electrical activity of these neurons, which represents their ability to communicate with each other, and the chemical activity, which is the actual language or the actual chemistry that comes out of neurons after they've been electrically activated. And while some great tools exist to study the structure and the electrical activity of neurons, the chemical activity of neurons has been known as this missing dimension in neurobiology, especially when it comes to neuromodulators such as dopamine, serotonin, and norepinephrine that are very difficult to image. Specifically, the electrical activity of neurons and the chemical activity of neurons is not a one-to-one relationship, as some recent studies have suggested. Therefore, developing tools to study the chemical activity is of paramount importance to get a complete picture of how the brain works. And this is especially important because neuromodulators are at the core of many of our psychiatric and neurodegenerative diseases. So being able to image them will give us a much better handle on the study of these diseases. So my lab has used single-wall carbon nanotubes, which is a semiconducting material, for the purposes of developing these probes. The way that we develop these probes is by taking these materials, which are intrinsically fluorescent, and modifying them with an amphiphilic polymer. This is a polymer that will adsorb onto the surface of the carbon nanotube, and through screening, we can identify polymer nanotube complexes that will selectively change their color or change their intensity in the presence of a particular analyte of interest, such as a neurotransmitter. And so by doing so, we've discovered some probes for neuromodulators such as dopamine, which we've excitingly validated in living brain tissue. The experiments that we do to validate the ability to image dopamine is by taking a mouse brain, making coronal slices or acute slices from this tissue, and incubating this tissue with our nanosensor. The nanosensor is then taken to be imaged, and we can stimulate the brain tissue at various different amplitudes, allowing us to see that as the fluorescence increases with this tissue, or as the stimulation amplitude increases with this tissue, that we get a commensurate increase in the fluorescence as well. Now, one of the exciting aspects of this probe is that it is also compatible with dopamine pharmacology. And so what this means is that we can use this probe to study the effects of dopamine targeting or dopamine receptor targeting drugs. One example is sulpiride, a D2 receptor antagonist, which would increase synaptic dopamine, or quinprol, which is a D2 and D3 receptor agonist and would have the opposite effect in decreasing synaptic dopamine. And so the assay that we can do now is taking our brain tissue, which is labeled with these nanosensors, and stimulating the tissue either in the absence of drug, as is shown by this flag trace, or in the presence of quinprol, which almost completely abolishes the dopamine release response. We can wash out the drug and confirm that this also recovers back to the original fluorescence. Now, one of the advantages of having a nanoscale tool is that we can visualize and see micron level features of these responses and identify hotspots that are putative dopamine release sites. If instead of ensemble averaging over the entire data, as we have done in these top traces, we now ask what each of these putative dopamine release hotspots is doing, what we see is that there's actually quite a lot of heterogeneity in how these hotspots are responding to drugs. Where here, if we look at the fluorescence response ratio, 1 being no effect to drug, less than 1 being a decrease in synaptic dopamine, and above 1 being an increase of synaptic dopamine, what we see is that while most of these dopamine release sites are actually responding to the drug as expected, which is a decrease in the fluorescence output, certain hotspots are actually having the opposite effect. And so this hints to either biological or drug-based variability that could be affecting how our pharmaceuticals are interacting with our neurons. Lastly, the lab is also really interested in making these developments more high throughput. And we've recently developed a probe for an analogous neuromodulator, serotonin, which has implications in maternal weaning. And are excited to get back into the lab and start using this neuromodulator probe for commensurate neurobiological studies. So with that, I would greatly like to thank the individuals who did the work. As I mentioned before, the work was largely done in the plant science space by Guzda Demer and Huan Zhang, and also the neurobiology work and the neuroimaging work largely developed by Abraham Bayene and Sang-Hua Zhang. I would especially like to thank the support of the Beckman Young Investigator program. That was the very first extramural source of support that my lab has gotten, and I don't know if these bold ideas would have gotten off the ground without that early source of support. Lastly, I would also like to thank and acknowledge Abraham Bayene, who developed the dopamine imaging probe. Abraham started his lab at the HHMI Genelia Research Campus earlier this year. And so if you are a trainee in the audience and interested in the neuroimaging line of work, I strongly recommend you reach out to Abraham. Similarly, Guzda Demer is the graduate student in my lab who developed the plant transformation tools. And so if you're a trainee in the audience and interested in the plant genetic transformation line of work, I strongly recommend you reach out to her to inquire about graduate student or postdoctoral opportunities. Thank you for your attention.